Hello students, welcome to EPG Pathshala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Today we are going to discuss about a module on fate of glucose under paper Carbohydrate Metabolism. So students, let us see what we are going to learn in this module. We will try to understand the fate of glucose under different conditions. We also try to understand how glucose transport and undergo phosphorylation. We will also understand the utilization of glucose, various pathways, and to understand the regulation of glycolysis. We know that each class of biomolecule has alternative fates depending on the metabolic state of the body. The cellular fate of glucose begins with glucose transport and phosphorylation. Subsequent pathways of glucose utilization include aerobic and anaerobic glycolysis, glycogen formation, and conversion to other intermediate in the hexose phosphate or hexosamine biosynthesis pathways. The intracellular form of glucose is glucose 6-phosphate. Only liver cell have enzyme glucose 6-phosphatase that dephosphorylates glucose 6-phosphate and release glucose in the blood for use by other tissue. Glucose 6-phosphate can be oxidized for energy in the form of ATP and NADH. Glucose 6-phosphate can be converted to acetyl-coenzyme A and then fat. Excess of glucose 6-phosphate is stored away as glycogen. Glucose 6-phosphate can be shunted into pentose phosphate pathway to generate reducing power, that is NADPH and ribose 5-phosphate. Glucose transport and phosphorylation. Each class of biomolecule has substitute fates depending on the metabolic position of the body. Glucose, the glucose 6-phosphate is intracellular form of glucose. Glucose released in blood which further used by tissue and cell when glucose 6-phosphate dephosphorylate by glucose 6-phosphatase. The ATP and NADH is released when glucose 6-phosphate is oxidized. Glucose 6-phosphate can be transformed to acetyl-coenzyme A and then fat. Excess glucose is stored as glycogen. Glucose 6-phosphate can be shunted into the hexose monophosphate pathway, release NADPH and ribose 5-phosphate. Glucose utilization pathways. Pyruvate oxidation, which may undertake more oxidation in the TCA cycle. For rapid utilization, store as the polysaccharide glycogen and renovation of glucose to other sugar and intermediates necessary for other important biosynthetic metabolic pathways. It can use for production of glycerol 3-phosphate and glycerol 3-phosphate convert into triglyceride and phospholipid. In body, a main cellular fate of glucose found in such tissue like adipose, muscle, and liver tissue. For the ATP generation, glucose is more efficient than fat and other fuel sources. Additionally, glucose is also produce ATP in an anaerobic condition. Figure shows the glycolysis pathway. We know that when glucose 6-phosphate, it converts into fructose 1,6-phosphate through fructose 6-phosphate as an intermediate. The first reaction is reversible. So during the first phase of glycolysis, the products are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which in turn in second phase channelize to produce pyruvate at the end. As we discuss that the glycolysis has two phases. First stage 1 to 5 step, phosphorylation occurs and glucose convert into two molecule of triose, glyceraldehyde 3-phosphate, and in the step energy, that is ATP, is consumed. In stage 2, which include step 6 to 10, pyruvate released from glyceraldehyde 3-phosphate through different 4 steps, and in this 4 step, during this step 4 ATP are released. It means that the net benefit is of 2 ATP. Glucose, if you see in the step first, that is a synthesis of glucose 6-phosphate. Glucose phosphorylated by enzyme hexokinase in presence of magnesium in the form of glucose 6-phosphate. In fact, all cells contain hexokinase enzyme, which is catalyze the phosphorylation of other sugar form such as D-glucose, D-mannose, and D-fructose. In fact, all cell, as mentioned, contains hexokinase. The phosphorylation of all the sugars is the base for the synthesis of ATP. You can see in the figure that during this stage, ATP is converted to ADP and 1 inorganic phosphate. Step 2, conversion of glucose 6-phosphate to fructose 6-phosphate. An aldose converted to a ketose. It's known as isomerization reaction. The glucose 6-phosphate and fructose 6-phosphate, both in cycle form. The reaction require ring opening and subsequent ring closing. Step 3, phosphorylation of fructose 6-phosphate. During this step, fructose 6-phosphate is converted into fructose 1,6-bisphosphate by enzyme phosphofructokinase. Here, ATP is converted to ADP and 1 inorganic phosphate. Step 4, splitting of 6-carbon compound. Aldolase enzymes catalyzes reaction 4 of glycolysis. The two triose, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, is released when fructose 1,6-bisphosphate is cleaved. Step 5. Interconversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Interconversion of glyceraldehyde 3-phosphate into dihydroxyacetone phosphate. During this step, the glyceraldehyde 3-phosphate undergoes oxidation and phosphorylation, and this molecule contains high energy which is released in form of ATP. An inorganic phosphate attachment consumes energy which is intake form in redox reaction. Step 7. Phosphoryl group transfer. During this step, phosphoglycerate kinase released inorganic phosphate from 1,3-bisphosphoglycerate and is converted into 3-phosphoglycerate. Step 8. Phosphoglycerate mutase. During this step, the 3-phosphoglycerate is not contain energy. For the ATP generation, 3-phosphoglycerate rearranged in the form of 2-phosphoglycerate and is able donate its phosphate group to ADP. During step 9, phosphoenol pyruvate has a higher phosphoryl group transfer potential than does glycerate to phosphate because it contains an enol phosphate group instead of a simple phosphate ester. Ketone form is more stable than enol form. Here the ketone form is pyruvate while enol form is phosphoenol pyruvate. Step 10. This reaction is the last step of glycolysis. Pyruvate kinase remove inorganic phosphate from phosphoenol pyruvate. 2 molecules of ATP are formed from each molecule of glucose. Thus, aerobic glycolysis oxidizes 1 molecule of glucose to 2 molecules of pyruvate. NAD plus is generated by electron transport chain. Under anaerobic condition, pyruvate is reduced to lactate for generation of NAD. Phosphofructokinase is the key regulatory enzyme. It catalyzes the committed step of glycolysis, conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. It is activated by fructose 2,6-bisphosphate and inhibited by citrate. The level of fructose 2,6-bisphosphate, the allosteric activator, are regulated by a bifunctional enzyme with phosphofructokinase 2 activity and fructose bisphosphatase activity. Dephosphorylation of the bifunctional enzyme activate the kinase but inactivate the phosphatase. This lead to higher level of fructose 2,6-bisphosphate and activation of glycolysis. Following shows the glycolysis under aerobic and anaerobic conditions. As we know that the fate of glucose during glycolysis is to form 2 pyruvate molecule through 3 successive reactions. Now the pyruvate formed during glycolysis has different fate under different condition. Under anaerobic condition, it forms ethanol, and that happens fermentation to alcohol in yeast. Again, under anaerobic condition, two lactic molecules are also synthesized. That is fermentation to lactate in vigorously concentrating muscles, vigorously contracting muscles, erythrocyte, some other cell, and in some microorganisms. Under aerobic condition, pyruvate undergo oxidative decarboxylation and form two acetyl-CoA molecules, which in turn enters in the TCA cycle. Lactic acid fermentation. Aerobic reaction. During aerobic reaction, glucose converts into 2 carbon containing pyruvate in presence of oxygen molecule, and then oxidize into carbon dioxide and water. This reaction releases more energy than anaerobic reaction. For the production of ethanol in yeast, anaerobic condition must be required. Conversion of pyruvate to acetyl coenzyme A. In presence of glucose, in presence of oxygen, the glucose is completely oxidized into carbon dioxide and water, as we discussed, and it takes place during various processes like citric acid cycle and electron transfer chain. Pyruvate dehydrogenase is converts pyruvate into acetyl coenzyme, and the TCS cycle is start with acetyl coenzyme A inside the mitochondria. When pyruvate convert into acetyl coenzymes, the reaction is catalyzed by a complex enzyme system. Carbon dioxide is released and coenzyme A is added. ATP derived from glucose is shown in the figure. When glucose converted into 2 pyruvate during the glycolysis, you can see 2 ATP and 2 NADH are produced, and total ATP produces 6. 2 pyruvate when converted into 2 acetyl coenzymes, 2 NADH is the product and 6 ATP are produced. When acetyl coenzymes enters into TCA cycle, at the end of TCA cycle, 2 GTP, 6 NADH are generated and 2 ATP and other 18 ATP molecules are produced. Regulation of glycolysis. The glycolysis pathway regulate by 3 ways: at hexokinase, then allosteric inhibition, and hormone. Hexokinase. 3 type of hexokinase: hexokinase 1, hexokinase 2, and hexokinase 3 are found. Hexokinase is highly sensitive towards glucose concentration in blood. Glucose kinase found in liver as well as in certain cell in pancreas, intestine, and brain require much higher glucose concentrations for optimal activity, and it is not inhibited by glucose 6-phosphate. Regulation of glycolysis. The glycolysis pathway regulate by 3 ways: at hexokinase, then allosteric inhibition, and hormone. Hexokinase. Three type of hexokinase: hexokinase 1, hexokinase 2, and hexokinase 3 are found. Hexokinase is highly sensitive towards glucose concentration in blood. Glucose kinase found in liver as well as in certain cell in pancreas, intestine, and brain require much higher glucose concentrations for optimal activity, and it is not inhibited by glucose 6 phosphate. Allosteric regulation. Allosteric regulation of the enzymes that catalyze the three irreversible reaction, that is hexokinase 1, hexokinase 2, and hexokinase 3, phosphofructokinase 1, and pyruvate kinase. Pyruvate kinase and phosphofructokinase 1 are inhibited by high ATP concentration as shown in the figure. Hormonal regulation. Peptide hormone are also regulate glycolysis like glucagon and insulin. Pancreatic alpha cell produce blood glucose level is low, also inactivate pyruvate kinase. Pancreas beta cell produce white blood cell glucose level is elevated, glucose transferred to the cell surface by insulin and sensitive glucose transporter. So students, let us summarize what we have learned in this module. Brain depends upon glucose as its main source of energy, and tight regulation of glucose metabolism is very critical for brain physiology and other metabolic function in prokaryotes. Consistent with its critical role for physiological brain function, disruption of normal glucose metabolism as well as its interdependence with the cell death pathways form the pathophysiological basis for many brain disorder as well as other disease. Glucose homeostasis is fundamental for life and long-term health. As the most effective organ for insulin-stimulated glucose uptake in the body, the skeletal muscle is the key element for maintaining effective glucose homeostasis in many chronic diseases. Prevention of muscle mass in obesity, insulin resistance, or type 2 diabetes is important for effective treatment and presentation of this disease. As quoted by Arthur Schopenhauer, all truth passes through three stages. First, it is ridiculed. Second, it is violently opposed. And third, it is accepted as self-evident. In nutshell, even in a state of starvation, blood glucose is maintained right up until death. What this really shows is that even if you are starving and eating no carbohydrate or fat or protein, there is no such things as glucose deficiency. The body can easily make what it needs. So students, this is all about fate of glucose. Thank you.